Good morning, class. I hope you guys all had a good weekend. So let's go ahead and get started as we have a lot of material to cover today. I'm your instructor for this class, Professor Viscara, and I will be teaching you cellular basis of life. And today's lecture, we're going to be talking about eukaryotic cells. So a general outline for this class today. I'm going to do a little bit of introduction about myself. I'm going to go ahead and talk about both the lecture and lab syllabus, as I'm in charge of both. And I'm going to go ahead and give you an introduction to both pro and eukaryotic cells, and then go over some of the key organelles and their function. If you guys have any questions during the time, feel free to go ahead and unmute yourself or add it to the chat. But if you can, save your questions to the end, because more than likely I'll probably answer your question. So a little bit about me. I stand before you guys today because of these people right here, my grandparents. They immigrated from Mexico in the 60s to create a better life for their subsequent generation families. And because of them, I'm here today. And I thank them for all of the stuff that they've done for me. And I owe them a lot of gratitude for where I am in my life. I'm also a first generation college and graduate student. So if you guys ever have any questions about navigating your future endeavors afterwards, feel free to hit me up during office hours. So my basic background and training. So I got my bachelor's in science from UC San Diego. And I majored in biochemistry and cell biology while I was there. I'm currently in my last year of the biomedical science PhD program here at UC Riverside. And I'm hoping that by me teaching this class will be a stepping stone for me to be a professor at UCR. So my research specialty, so I have extensive research experience, and I have a few publications in these different aspects. So I'm well versed in cancer biology. The bulk of my work, which we'll be covering later in the class, is viral infections and immunity. A lot of my work had been focused on macrophage infection of dengue and Zika. And my current work is focused on the adaptive immune cell T cells that are recruited to the brain during toxoplasma infection. So if you guys are interested in hitting me up about any of that stuff and you want to know more about it, feel free to ask me during office hours. So the purpose of me wanting to teach is because I want to help change our society. I feel like with all of the craziness that has happened in this last year, I feel like a lot of things need to be done. And I feel like what I can do to help change the system is by educating. And so here I'm going to be educating you. And I feel like this class is going to be focused on trying to teach you about relevant topics that you guys can take into your own lives. So the way I have this class structured is basically like 4 different units, I would say. So the first week is going to cover like key core concepts that any cell biology class is going to go over. So key organelles in a cell, how DNA replicates, and this concept of the central dogma of molecular biology, where DNA converts to RNA and makes protein. And then as the pandemic has really changed our society, I want to go ahead and give you a brief introduction to viruses, but with an emphasis on COVID. And so those classes are going to be more geared towards understanding how COVID spreads, how it gets into the cell, and how it's able to hijack the host cell machinery that we're going to end up covering during this week, and how it's able to replicate and the disease manifests itself. And then I'm also going to talk about another kind of virus called bacteriophages that infect prokaryotes. And for that class, I'll be talking specifically about those kinds of viruses, but what I want to make relevant is kind of linking that to how scientists are using some of those bacteriophages now, and what it means for the future when it comes to making designer babies. On Monday of July 5th, we're not going to have any class as it's an observed holiday. And that Tuesday, you guys are going to have your first midterm. And that'll be offered during our normal 9:30 to 11. So after that midterm, basically, I'm going to focus on the immune system. So I'm going to break it up into two sections, basically focusing on key innate immune cells. So the first line of our defense when it comes to fighting against any pathogen, and highlighting a couple of key immune cells that are implicated in recognizing the virus. And then after that, I'm going to go ahead and talk about the adaptive immune system, which is basically responsible for defeating the virus. Once we have a general understanding of how the immune system works, I'm going to get into some tools for both testing and defeating the virus. So for the testing, I'm going to be focused on antibody production, which I'll cover somewhat in the adaptive immune system section. And then also something called polymerase chain reaction. And that'll be important when we're talking about how DNA replicates and how scientists are able to use some of that to detect coronavirus. And then for class 12, I'm basically going to highlight a lot of the different tools that we now have after the pandemic has begun to really take care of this virus and flatten this curve. I'm going to highlight how vaccines work, the different kinds of vaccines that are out there currently on the market, how antivirals work, and some other therapies such as monoclonal antibody therapies and how those are implicated in trying to decrease the overall viral load in an infected individual. And then I'm going to end the class with the last unit talking about nutrition, because I feel like nutrition is a very important topic that we need to really understand. And with all the disease that happens in this country that's relevant to, too much sugar intake, diabetes, and cardiovascular disease, just because of a lot of fat in the diet, I want to talk about some of the macromolecules that are involved in that, but also link it to how the immune system responds to some of those molecules and how a lot of that helps the virus manifest stuff. Because as we know, a lot of patients that are most susceptible to succumbing to the infection are people that are diabetic, are hypertensive, and have some kind of cardiovascular disease. And so I feel like for you guys to have an understanding of how these diseases manifest and linking back that to COVID, I feel like that's an important takeaway from the class. I'm also talking about some important vitamins and antioxidants, and I'll be linking that again to the immune system and the virus. And then the last class is going to be focused on giving you guys tools on how to decipher good science versus fake news, because I feel like that's been a main topic in the media in this last year. And I feel like, including my family members, a lot of them don't know how to decipher what's fact and what's fake. And so that class, I really want to help you guys understand how to identify misinformation, as well as give you guys some sources for validating information that's out there. And then the final will be given on July 23rd, and that'll be given from 8 a.m. to 11 a.m. And so here is the overall course breakdown, both the percentage and the points. So in-class participation. So this is going to be worth 5% of your grade. And because it's part of your grade, I'm making class mandatory. I feel like there's a correlation with what your grade turns out to be, how often you go to class. And so I feel like giving you guys easy points to incorporate into your grade will hopefully get you guys to come to class more often. But more importantly, a lot of the reason why I want you guys to go is not just, simply to lecture, but also I have questions that are embedded within my lecture that are going to be basically what I'm going to be asking on the exam. And so I want to know that you guys are actually understanding the material. And so what I'll probably end up doing is putting you guys into breakout groups. You guys work on the questions together, and then you guys come back, and then we'll go ahead and take polls to see how people are doing with the questions. because I don't want to find out on the exam day that everyone struggled with this concept and, no one really, gave me a heads up on that. So I feel like these in-class activities will help me gauge that. Another easy set of points are lecture quizzes. So at the end of every lecture, I'm going to go ahead and post on iLearn anywhere from 3 to 5 questions that are relevant to the lecture I had just gone over. And again, a lot of those questions are going to be exam style questions, and I might even recycle some of them. So please don't learn the material passively. You guys will have until Friday to answer all of those questions. So please get them in on a timely fashion. And you guys have 2 chances to take that quiz, and whatever your better score is, that's what's going to get recorded. So, most of you guys should be able to get perfects on those tests. And so the bulk of your grade, though, are going to be the exams. So we have a midterm that's going to be on July 6th, and it's going to be given during our normal class schedule. And you're going to have 80 minutes to complete that. It's going to be open book and open note. So I'm not expecting people to memorize the material I go over. I want you guys to understand the core concepts and some of the terminology, but I'm not expecting you guys to memorize a lot of stuff. There's going to be a lot of material we cover. But when I say open book, I mean, use your notes, use my lectures, use the lab worksheets that I'm going to go over in a bit, but this does not mean to Google answers. This isn't going to help you learn any of the information, and I view it as a form of cheating, so I will not appreciate that. So please learn the material. I make it very easy to learn. The questions are going to be straightforward. I'm not here to trick anybody. I want you guys to all pass and do well and really take away a lot of the core concepts that I go over. And then overall, the midterm is going to be worth 20% of your grade. Because it's only 5 weeks for the class, I didn't feel it would be fair to make 2 midterms. I think that's just too fast. So I think 1 midterm is going to be sufficient. And then the other bulk of your grade is going to be your final, which is worth 30% of the grade. And as I said earlier, it's going to be given on Friday, on July 23rd from 8 to 11. And you will have 3 hours to complete this. And just like the midterm and basically all the quizzes and stuff I give you, it's open book and open note. I think that's all for that. So going into the lab. So the labs that I have are going to be very straightforward. You guys are all enrolled in a section that's offered Tuesday and Thursday, so it's going to be twice a week. And all my lab modules are going to be based off of some important concept that I really want you guys to understand that I covered during that week. And then there's going to be 2 assignments that are due. You're going to have a pre-lab that's due before you come to class. So there's one currently posted. So please get that done before your class tomorrow. So it's either 1 p.m. or 4 p.m. And then there's going to be a worksheet that's going to be the majority of your lab grade. Your overall lab grade is going to be worth 30% of your grade of your overall biology 2 class. So please, those are easy points. Try to go ahead and get all of that in a timely fashion. I see a question. So will there be 4 quizzes due on Friday? Yes. So there is going to be 4 quizzes for the lectures that are due on Friday. However many lectures there are. So I know in week 3, there's like 2 lectures we only have, so it'll just be those 2. But yeah, so they'll be due at 5 p.m. on Friday. And I believe that's all written in the syllabus. And also when you're taking the quizzes, I have that information as the instructions. So if you guys forget, it's posted there, or if when in doubt, ask me. So the pre-lab quizzes. So again, you guys have one due tomorrow. You guys will have one due always before the start of your lab class. So that's going to be 2 a week. And again, it's due at the start of whenever your lab is due. I will make these quizzes available at least 24 hours before they're due. I'll probably do a little bit longer just because I know some of you guys work and it's really difficult to, plan accordingly. Um, but yeah, the way that these pre-labs are going to be is basically I'm going to have you watch a video or two, and then that's just going to be followed by a quiz. And so the quiz is going to be very straightforward. It's also going to be multiple choice. You will have 2 chances to take it. So again, the better one is going to be what we record. And these pre-lab quizzes are worth 10 points each. There are 8 quizzes total, so that's going to be 80 easy points for you to not miss out on. So please, please do them. If you have a conflict for the final, please email me, and I will go. We can go ahead and schedule stuff accordingly. And yes, lectures will be recorded and posted on iLearn. So I usually am pretty good about recording ahead of time, or like pressing the record button. If for some reason you don't see something with me recording, please let me know. But I will always make a note to myself to make sure I record these, and I will go ahead and post them sometime later today once I have a chance to upload everything. So the overall lab course and its layout. So again, you guys are going to be going to lab sections twice a week, and these are going to be for basically 3 hours long. The way that it's going to go is your TA is going to give you some kind of short lecture to highlight some key concepts I've gone over during the week, and they're usually going to be relevant to whatever the lab worksheet is based off of. And then after, they're going to go and give you instructions on how to actually do the lab. I'm going to leave it up to the TAs if they want to do the questions all together as a class, or if they're just going to have you do it individually. But I'm going to give them that freedom. You will have access to the worksheets on iLearn in the assignment tab of your respective lab section. And those will usually be available to you around maybe about an hour or two before your class actually begins, if not at the start of the hour. So go ahead and do them in class. You have about 24 hours to submit your worksheet on, and all this is going to be on iLearn. So please get this done on time. You really don't need all 3 hours, at least the way that I'm structuring a lot of these worksheets. So get it done during that time. They're very straightforward. They're going to be there to help reinforce a lot of the biological concepts I go over, and you have your TA present to walk you through any questions you might have. Each lab is worth 30 points, so this is going to be a total of 240 points. So please submit everything on time if you can. If you miss that 24-hour deadline, you have time to still submit late assignments, but you're going to get docked 10% every late day, and that's going to be about 3 points missed out. So those are easy points, and those can add up if you're constantly missing assignments or turning them in late. So again, try to get these done while you're in class. I've also instructed the TAs to record their Zoom sessions. This is going to be both for the benefit of the student and the TA, and I've instructed them to upload them after their class is done. So for the lab worksheets, again, they're going to be an interactive worksheet. So it's either going to be a computer game or some related video or videos to reinforce the concepts that I covered. So for example, the worksheet that I have that's going to be due for this week for lab 1 is basically you're playing a computer game where you're traveling within a cell. You got to shoot a couple of organelles, and then you learn about what those organelles are, and then you're going to use that information to help you answer the questions on the worksheet. Very straightforward, very easy. No tricks or anything like that. If you have any questions about it, ask your TA, and then when in doubt, ask me. And again, they're due 24 hours. Try to get them done within that class. And then again, any question that I have or I have made, don't learn it passively. Actually learn the material, because some of these questions I will recycle, or I will ask something similar to it. So this will help you get a feel of what to expect for the exam. My absence policy. So again, I'm expecting you guys to come to both lab, which is mandatory, and lectures. I understand that some people work and stuff, or there's some exigent circumstance, such as a death in the family or some kind of medical emergency. I'm very understanding of this. Just if you know you're going to miss ahead of time, please, please, please fill out this absence form and email me immediately, because I want you guys to get your points for the day. And so basically what's going to happen is if you end up missing any of these classes, and you're really going to be limited to a couple that you can miss. And, you know, depending on your situation, I can be flexible. But the way it's going to work, if you're not going to be able to attend for some exigent circumstance, what I'll probably have you guys do is answer any of the questions that are embedded within my lecture, and then email me those answers that you get. And then you won't miss out on any points. But this is going to be offered only a handful of times, so don't make this a regular thing. And then, yeah, email me to alert when you submitted that form and if you have any issues ahead of time. Here's all of my contact information. So I have my phone number included on this, but please only text me or call me if it's an extreme emergency. But please use my email address as often as you can when it comes to contacting me. I've updated a new syllabus, which I think I sent out a message for all of that. So I've moved my office hours now to Thursday from 11:30 to 12:30, just because I think what I originally had cut into one of the lab sections. So come during then if you guys have any questions, or if you guys can't make that hour and need to meet me at a different time. I'm very flexible. So just, you know, communication, that's the key here. Is that everything about this? I think so. So the overall expectations from people, I expect everybody to treat every member in the class, so that's myself, the TAs, and all of your classmates, with respect and equality. If there are any issues that are going on amongst anybody, please contact me immediately. I have no tolerance for anybody disrespecting somebody else, and I will handle it. You guys are all expected to be on time and prepared to participate in both the lectures and labs, and you're supposed to go to your registered lab section every week. The TAs will be taking attendance, so if you can't make it, email both myself and the TA, and then fill out that excuse absence form. Turn in your assignments on time. These are all easy points. I give you ample time to finish your material. And so I feel like all of you guys should be able to do well in the class. If you guys are struggling with the concept, please come to my office hours. I'm more than happy to go ahead and walk you through some of the stuff, because some of it might get a little confusing. So, yeah. And then follow campus policies on academic integrity and honesty. I have no tolerance for any kind of cheating. So if it comes down to that, where it's learned that you are found cheating, depending on what it is, you're either going to receive a 0 or you're going to fail the class, and then there's going to be some additional meetings. So please, I don't want to deal with any of that. I give you guys all the material you guys need to pass the class, so don't do any of that. All right, so any questions about the syllabus? Okay. If you guys have some, feel free to shoot me some emails. So let's go ahead and talk some science now. So for a lot of my lectures, I'm going to be, let's see. What were office hours again? They're from 11:30 to 12:30 on Thursdays, and just use the same Zoom link. And then all of that material should be on my syllabus. So there's a link in there, and there's also the login information. So I'm going to have learning objectives. And so if you know what the learning objectives are, if you can answer a lot of these things, you're going to do fine on the exam. Again, there's no tricks or anything. I'm very straightforward. I'm going to tell you what I want you to know. Anything that's bolded in my lectures are things that it's probably a good idea for you to know, because I'm probably going to ask it. Anything that I emphasize or go over a lot, for sure, I'm gonna end up asking you questions. So please focus on those things. I understand there's gonna be a lot of material to cover, but I'm gonna make everything fair. So don't worry. So today, the lecture is focused on eukaryotic cells, but I'm basically going to use prokaryotic cells as a way to compare the two types of cells. I really want you guys to know what do both of these cell types have in common, but also what distinguishes them. The main organelles that I want you to know in eukaryotic cells are the ones listed here. So the plasma membrane, nucleus, endoplasmic reticulum, Golgi apparatus, and mitochondria. And I'm going to end up going more in depth. So anytime I go into more depth of a topic, I'm going to end up asking something on it. So if I don't really touch it, it's unlikely I'm going to ask something, but anything's fair game if it's in my lectures. And then I want you to be able to use the analogy, a cell as a factory, because I think using that analogy will help you understand what the function of a lot of these organelles are, as there's quite a few of them. Question. Yes, the PowerPoints are posted on iLearn. I believe this lecture is already posted. If not, let me know. But I believe it's posted on the actual A01 iLearn section. And I believe it's, I forget what tab it's in, not course material. So yeah, I'll usually post them ahead of time. So yeah, go ahead and download them, take notes on there. I have notes already on there, but under my outline or notes section, I'll have some additional videos on there. You guys don't need to watch them, but if you guys are interested in learning about whatever the topic I'm presenting, those videos are really helpful and go more in depth. So yeah, so here I have an image of the hierarchy of life. And so we start with the biosphere and work all our way down to an atom. And so the scope of the class is really going to be at this level where we're talking about really the cell and all of the components that go into it. I will talk a little bit about some tissue, but that's going to be really when I'm talking about disease. But we're really going to be focused on the actual cell and all the biological components that are within it and how they function. Cells can be grouped into two categories: prokaryotic cells and eukaryotic cells. So prokaryotic cells are single cell organisms. So that's all they are. They're just one unit and that's all they are. And so two domains of cells that exist within the prokaryotic cell are the bacteria, and I have a picture of E. coli here, and archaea. And with the eukaryotic cells or multicellular organisms, there are 4 different domains. So there's animals, so we're animals. I have a picture here of a lung section, so you can see the complexity of basically the lung tissue and all of our airways. We look at plants. Here's a cross section of a fungus or a mushroom, also multicellular. And then we also have this other group called protists. And so things that fall under protists are like amoeba and diatoms. And here's a picture of a diatom. And fun fact, diatoms they're found in the ocean, and they are responsible for producing at least 25% of the oxygen that we breathe. So it's really important to really, keep our oceans clean. So again, prokaryotic cells are single cell organisms, and bacteria and archaea fall under this. The fossils that we have for prokaryotes date back all the way to 3.5 billion years ago. So this is about 1 billion years after the formation of Earth. So what I'm going to want you to know with a lot of these are some of the unique features of bacteria. And so here I have highlighted in blue what is specific to only eukaryotic cells. And so when we're comparing different organisms together, I'm really going to be having you compare bacteria, animals, and plants to one another. So don't really worry about any of these other groups. So for bacteria, you basically have four unique components. So we have the capsule, which is basically composed of a bunch of different sugars that help the bacteria stick to surfaces. We have pili, which there's these projections that are coming off of the bacteria. And there's two kinds. So there's an attachment pili, which, as its name implies, will help it attach to surfaces. And then we have sex pili, which is how bacteria are able to exchange genetic information with each other. So basically their version of sex. And so here we have two bacteria that are connected through this pili exchanging genetic information. We also have a nucleoid. And so a nucleoid is basically the bacteria's DNA. Think of it as their own chromosomes, but it's different from ours in the sense that it doesn't have a membrane that protects it. It's basically free floating within the actual bacteria. And I'll get more into what I mean by the membrane when we're talking about eukaryotic cells. And then we have these other short rings of DNA called plasmids. And so what these plasmids usually are, so they're different than the nucleoid because they're not really, the nucleoid has information that's going to encode how the bacteria is. So like how its cell wall is composed, all the proteins that are involved in making its structure is all encoded in the nucleoid. With the plasmids, they're actually very short sequences of DNA that encode for things such as bacterial resistance, or antibiotic resistance. So basically when there's genetic mutations within a bacteria and they're able to basically survive from antibiotic treatment, the reason why they're able to do that is because they have a sequence in here that allows them to do that. And that's usually passed during this process of conjugation. So what I have highlighted here in magenta are features that are found in both eukaryotic and prokaryotic cells. So here bacteria have this, some species of fungus, and the only animal cell that I'm aware of, or at least in humans, are sperm. And so the flagellum, the whole purpose of it is to help aid in movement when the organism is, or the cell is traveling. In green, I have highlighted basically what's found between bacteria, some species of fungi, and plants. And so the cell wall is just going to be a rigid structure that keeps it intact. And then in red, I basically have components that are found in really all sources of life. We have ribosomes, which I'm going to get more into later, that are going to help facilitate building proteins. We're going to have a plasma membrane, which regulates what goes in and out of the cell, such as water and ions. And then a cytoplasm, which is a jelly-like substance that kind of keeps all of these materials within a cell suspended. Are plasmids small copies of the nucleoid? No, so they're separate from it, and I'm not really sure the process on like how these plasmids are actually synthesized. I could definitely look into that if you want me to. If there's a question I'm not sure of, I can always answer later. So yeah, I think that's everything for this slide. So what are you really made out of? So did any of you guys know that scientists estimate that only half of the cells in the body are actually human? The rest are actually bacteria. And a question that I want to ask, is there such thing as good bacteria? And so I'll give you guys all a quick second to answer. All right, I think that's almost everybody. I'll give you guys another couple seconds. All right, I'm gonna end this. So good. I'll share the results with you guys. The majority of you guys got that there is such thing as good bacteria. And so what these bacteria do for us is protect us from foreign pathogens, such as bad bacteria or viruses or parasites. And so the location that we have on our body of bacteria are on our skin. So that's one of the lines of defenses for preventing bad bacteria from growing on us. And the other source that we have is also in our mouth, because we don't really want any other bad bacteria growing in there. That's going to result into an infection. The last place where the majority of this bacteria live are actually in the intestine. And so what they're called is a microbiome. And it's really important to really keep your microbiome healthy, because these bacteria are actually really important for making things such as vitamin K and some of our energy that's derived from short fatty acid chains. So yeah, it's really important to really keep that happy biome. And I'll probably talk a little bit more about this in a separate slide or in a separate lecture. So how is this possible? Well, it has to do with the size of the bacteria. So if we look at the scale, we have a strand of hair at this end, and then on the bottom side, we have a viral particle. And so when we look at bacteria, we see that it's roughly 1 to 3 microns long. When you compare this to a human cell, here we have a red blood cell or an immune cell. This one. It's basically at least 10 times smaller. So basically, if you're comparing it to volume, for every 1 human cell, there's 10 bacteria that are able to take up that same volume. So that's how bacteria are able to outnumber us based off of cell number. I got a couple more questions. Oh, okay. There's some other stuff. Okay. So eukaryotic cells all contain organelles. They are much bigger than prokaryotic cells and are much more complex. So a lot of eukaryotic cells within them have these membrane-bound organelles. And I'm going to go ahead and highlight a couple of them that I want you to know that are within this lecture. So all eukaryotic cells are fundamentally alike. They share a lot of the same structure and a lot of the same organelles that have the same function. So the main ones that I want you to know that they have in common are this plasma membrane, the nucleus, and the mitochondria. And I'm going to go ahead and talk a little bit about more each one of these. So here is a structure of an idealized animal cell. So it kind of has everything that you would see in an animal cell. Again, flagellum, the only animal cell that I know that has it is sperm, but I think they included this for teaching purposes. So by the end of this class, or at least this end of the week, including the worksheet that you guys are going to do, you're going to learn about a lot of these organelles, but the ones that I cover in this lecture are going to be the ones that I really want you to know. So for this slide, what I really want you to understand is that lysosomes are, the only organelle that is unique to animal cells. So if I ever ask you a question like, what is unique to animal cells? And I give you a list of a bunch of different organelles, lysosomes would be the answer. So understand some of these key differences between them. So, and then here we have a structure of an idealized plant cell. So what we have in green are specific structures that are unique to plant cells. So again, understand that these are, if I ask you a question like what's unique to these, know that these will be the answer. So here we have chloroplasts that are basically how photosynthesis is able to be done. You have a central vacuole that holds a lot of the cell's water. And then you have the cell wall that kind of keeps the cell wall of a plant kind of intact and connected to one another. So every cell is surrounded by a plasma membrane. So that's both pro and eukaryotic cells. And so the whole point of this plasma membrane is to regulate what comes in and out of the cell. And so the main components that the plasma membrane regulates are ions such as sodium, chloride, and potassium, sugar, and water. And so a lot of these are going to facilitate how the cell size is. So water and ions usually play a role on shrinking or expanding a cell. And then the sugar is usually going to be a nutrient for the cell to actually function. Here we have a structure of what the plasma membrane looks like of a typical cell. So here we have the inside of the cell on the and here we have the outside. And so with on the plasma membrane, we have typically two kinds of proteins. We have a membrane protein that is called a receptor. And we'll get more into what receptors are when we're in the immunology unit. But basically what receptors are is something is going to bind to the receptor and then some kind of signaling transduction mechanism happens. The easier are these other membrane proteins called transporters. And just like the name implies, it transports things in or out of the cell. I'm not going to go into the different kinds of transporters that exist. But here we have an example of a sugar transporter. Basically, sugar is going to come from the outside environment and then bring the sugar into the cell so it could be used for nutrients. And then the majority of the plasma membrane is made up of these phospholipids. And so there's going to be two layers of them, so a phospholipid bilayer, and they're consisted of two parts. So they're going to have a phosphate head. And so this is going to be the part of the molecule that interacts with water. So it's water loving, so hydrophilic. And then what's going to be facing on the inside of the bilayer are these hydrophobic or water-fearing fatty acid tails. So if we go back to looking at the structure, we have these two rows where the phosphate heads are basically interacting with the watery environment, and the hydrophobic fatty acid tails are facing inside. So for those people that want to know about the structure of it, I have an image here of what the phospholipid head consists of. So it's just a phosphate group and a glycerol, and then the fatty acid tail is just these chains. And so I'm never going to end up asking you like what a structure is or have you draw something. But what I do want you guys to know is if I have a picture of a phospholipid, I would ask you a question like which part of it interacts with water, which area is hydrophobic or something to that nature, just to understand the concept of what are the key components to what a phospholipid is. And so within eukaryotic cells, we have these membrane-bound organelles. And so here I have listed of what's found in plants and what's found in animals and what they have in common. And so with plant cells, what's unique to them are these plastids. And so the plastids are the chlorophyll, so the organelles that are meant to do photosynthesis. And then they also have the central vacuole that stores all of its water. Unique to the animal cell are these membrane-bound organelles called lysosomes. And what these lysosomes do are basically these vesicles that within them have a really high pH, or it's actually low pH, sorry, so it's more acidic. And what the whole function of them is to degrade things. And so typically a protein that wasn't folded properly will go to a lysosome so it can get degraded. An intracellular pathogen will get sent to a lysosome so it can get degraded as well. or other components that a cell uses so it can recycle materials such as nucleic acids and other kinds of proteins. But what they have both in common between a plant and animal cell are a nucleus, endoplasmic reticulum, a Golgi apparatus, and a mitochondria. So here I have a list of the organelles that have a single membrane versus double membrane. So basically the single membrane organelles are going to be the lysosomes, the endoplasmic reticulum, and Golgi apparatus. And this is a general structure of what they look like. I'm going to go more into depth about these two. And then the double membrane organelles are going to be the nucleus, which houses your DNA. And then these two other organelles, chloroplast and mitochondria, that are responsible for creating energy for the cell to function. And I will also be going more in depth into what these are. So let's start off with the nucleus. So the nucleus, again, is a double bounded membrane organelle. And this is where your DNA is stored at. So all your chromosomes, all your genetic information, all the things that make you, you are stored in here. And so if we use the analogy of a factory, think of the nucleus as the central office of the factory. Again, to reiterate, bacteria do not have a nucleus. They do not have this membrane-bound organelle that protects its genetic material. That's nucleus is specific to eukaryotic cells. So again, the nucleus is where your DNA is stored. And so chromosomes, all they are is just tightly wound DNA. And so if you think of DNA, it's actually very long, but in order for all of your genetic information to get stored within your cell, it has to be really condensed. And so the way it's able to do this is by these proteins called histones. And so what ends up happening within the cell is DNA wraps around these histones, and those two together are, for terminology's sake, are called nucleosomes. And so these nucleosomes are coiled together and they form these chromatin fibers, and then together those get supercoiled and get really condensed and make up your chromosomes. So typically we think of DNA as this double helix structure, which it is, but when we're looking at the structure of where it is within the chromosome, it's actually really tightly condensed. So I got a question: if bacteria don't have a nucleus, where is their DNA stored? So so if we go back here, it's the DNA. So this is their DNA right here. So instead of you know, them having a nucleus where the DNA is stored within, they just have their DNA floating within them, and it's just all compact as this image is shown here. So it's in this case it's called a nucleoid, but it doesn't have it has its DNA, but it doesn't have a structure that's protecting it. All right. So let's see how we're doing on time. Okay, I got some time. So so again, the whole purpose of DNA is to make proteins, and it's a very important process. So this you know, we're all protein-based organisms. Any living being or you know, cell, all are made up of proteins, and then we have some sugars and fats that get added on later to the protein. But this is done by this concept of the central dogma of molecular biology. And all this means is the DNA gets converted to RNA, and that RNA gets converted to protein. And I'm going to be talking a lot more about this in lecture 3, but I'm just giving you guys a preface of what this consists of, just so I can go in and layer on a lot of these concepts, because this process does get complicated. So just to kind of give you guys a preface to this, the name of the process between DNA converting to RNA is called transcription, And the process between RNA converting to protein is called translation. And so there's several organelles that are involved in making a protein. And these are the organelles that I really want you to know and highlight. So when we're talking about transcription, where we're converting the DNA to RNA, this all takes place within the nucleus. Once the RNA is synthesized, it's going to leave through these nuclear pores that are on the nucleus. So again, the nucleus is just a structure that houses the DNA. It's not actually the DNA. And then for the process of translation, basically there's going to be a couple of different organelles that are involved in this. So the RNA is going to get exported out of the nucleus, and then it's going to get sent to the rough side of the endoplasmic reticulum. And so on the RER is going to be studded with these ribosomes. And you have two kinds. You basically have kind that are with on the actual structure, or you have some that are floating within the plasma of the cell. But this is where translation takes place. So this is where the RNA gets converted into a protein. But once the protein is synthesized, it actually has to be folded and packaged properly. And so after that, it's going to end up getting sent to the Golgi apparatus, where after it's folded properly, you can get shipped off to wherever the cell needs it. So here's just another way of visualizing this process. We have the DNA that converts to RNA under this process called transcription. The RNA leaves the nucleus, where translation can take place at the site of the ribosome. The ribosome will synthesize the protein, and the protein will get sent off to the Golgi apparatus, where it can be folded properly and sent off to wherever it needs to go. So just you're going to hear me repeat a lot of the stuff, and hopefully by the end of it, a lot of it will make sense. But again, the stuff that I emphasize are things that I'm going to end up asking you about. So again, ribosomes is where protein synthesis begins. There are the ribosomes that are bound to the rough ER, and then there's ribosomes that are, they call them free ribosomes, that are just floating within the cytoplasm. Depending on where these ribosomes are located will determine where the finalized protein gets sent. So a lot of the stuff a lot of the proteins that are synthesized at the rough ER, those proteins are going to end up really being sent to the smooth side of the endoplasmic reticulum, the Golgi apparatus, lysosomes, or get sent out of the cell or be part of the actual membrane of the cell. The free ribosomes, which again are floating within the cytoplasm, are those proteins are going to end up getting sent to the nucleus, probably to be used for the membrane structure. It'll be used for the mitochondria, peroxisomes, which I'm not going to get into, or other cytoplasm components. So again, with the endoplasmic reticulum, it has a smooth and rough side to it. For the smooth side located over here, basically this is where a lot of fats or lipids are going to be made for the actual cell membrane to keep its structure. And some of those fat molecules will be sent out of the cell. And then where the rough ER, that's where the ribosomes are at, where protein synthesis can begin. And so if you compare the endoplasmic reticulum to some kind of, or use the analogy of the factory, consider this to be the assembly line. This is where the proteins are being made, and then they're going to get finalized and packaged off to the Golgi apparatus. So the Golgi apparatus is where these proteins are going to be modified and finished so they can be sent off to wherever they need to go. So here's where typically the protein will be added with some sugar residues or some fat residues and folded to whatever structure it needs to look like, and then go to wherever it needs to go. And so if you compare this to the factory, this is where your shipping and receiving is going to be. So we have two organelles that help provide energy for the cell. We have chloroplast, which is found in some plant or all plants and some species of algae, and the mitochondria, which is found in both basically all eukaryotic cells. I think most of you guys have seen memes related to the mitochondria, where it's all known that it's the powerhouse of the cell. But they always ask, what is the powerhouse of the cell? But they never ask, how is the powerhouse of the cell? I have a lot of friends that aren't in science and ask them, what does it mean for the mitochondria to be the powerhouse of the cell? Well, what that means is it's synthesizing and making energy. And the currency of energy in a cell is this molecule called ATP. ATP is basically how the cell is able to do stuff that requires energy when it's functioning, say like when it's moving or doing other importing or exporting molecules. So we'll talk a little bit more ATP in another lecture, probably closer to the end unit. But for now, understand that ATP is what the cell uses for energy. And so the way it does this is by taking in sugar and the oxygen that we breathe. So we eat the sugar, we breathe the oxygen, and then cellular respiration happens. And during cellular respiration, so there's this whole long process where there's the breakdown of the sugar called glycolysis and the Krebs cycle. I'll probably touch a little bit about that when we're in the carbohydrates lecture, but I'm not going to expect you to know all of that stuff. Just for simplicity for now, understand that this is what a cell needs to use to undergo cellular respiration. And the byproducts of this process is CO2. So we breathe out the CO2, we breathe out some water, and then ATP is made so the cell can do energy. It's also important to note that it's not just animal cells that do this, it's plant cells. A lot of people forget this. People don't realize that plants also breathe, or they also use oxygen. And the way they're able to do this is by the process of photosynthesis. And so, as we all know, with photosynthesis, plants need sunlight. And this sunlight, there's a whole process where there's a lot of electron energy that's displaced, so energy can be used. Not going to go into that. That's beyond the scope of this class. But what you need to understand for this process of photosynthesis is the cell uses CO2, water, and sunlight to undergo this process. And then from this process will yield the byproduct sugar and sugar and oxygen. And if we go back to this, sugar and oxygen are the product or the reactants that are needed to undergo cellular respiration. So basically, plants make a lot of their own oxygen and sugar in order to use energy for to create ATP. So it's an important concept to really understand, because I think a lot of students forget that plant cells also have mitochondria that use energy. And only plants do this, or anything with the chloroplast. So here I have basically the cell factory as summary. This will be very helpful when it comes to studying. I'll let you guys go ahead and go over it on your own when it comes to studying highlighting these key organelles that I really want you to know and understand the analogy between the factory and how a cell organelle functions. So if I were to give you a question like, using the analogy of the factory, which one of these organelles function as the power plant? And then I'll give you a list of the organelles, and then you pick the correct one. Let's see, I got some questions in here. Do plants have chloroplast only, or a mix of mitochondria and chloroplast? They have both. So let's just a nice image that I had here. So if we look here, we basically have two plant cells, and then this diagram highlights where the chloroplasts are and where the mitochondria lie. So plant cells have both whereas animal cells only have mitochondria. So there is some time right now. So I would like to break you guys all out into breakout groups. And I'll probably put you guys into a group of like 2 or 3. And so what I want you guys to do for about 5-ish minutes, pick your favorite organelle or an organelle we went over, describe its function and its relation to a factory, and explain why you like it. And the whole point of this is to really help you guys start processing the information I'm giving you guys in a different way. Studies show that the more times you process something, so that's from you hearing it from me, you explaining it to a classmate, you reading the material on the lecture slides, you seeing the questions on the worksheet and the lecture quizzes, those are going to help promote retention of the information. So when you see it on the exam, you're not trying to search for the answer, you already know it. So let me go ahead and I'm going to stop sharing this. And then after I'm done, I'll go ahead and give you guys the link to the sign-in sheet so you guys all get credit for attending. And that is the first lecture. Let me stop sharing this and then I'm going to go ahead and where is breakout rooms? Sign automatic. All right, I'll see you guys in a second. Open all rooms. Did you guys have trouble getting into the assigned one, or was there not enough breakout rooms that I made?